So the benefits to use this particular software, which is why we're also promoting it a little bit, is that it doesn't require uh, okay. It doesn't require any direct coding. So if you do not come with a computational background, do not know how to write codes, it comes with a very graphical interface. So there are icons that mean something. I'll tell you that they have a certain functionality, and those functions are pre-coded in the software. So, sorry? Sort of. Yes, it is like that. A little bit like that. Also, but a little much more varied, and there are ways in which you can incorporate conditions and more randomizations, which is built in within the experiment builder. So it offers you a range of possibilities that you want to design between tasks. Also, it's not only specified to writing experiments. You can make any sorts of experiments. It can be manual reaction time for EEG or for whatever. It just integrates well with the iLink system that SR research, which makes SR's experiment builder and iLink together. It does it very flawlessly. So for the ease of incorporation, experiment builder is quite convenient. Alright, so before we start the designing process, I wanted to take an example of a research paper. So often the problem that most people face even after knowing a technology is to how to start, how to approach an experiment design. You might have an idea, you don't know how to translate it into an eye tracking experiment. And we were in the question answers, we were discussing that just measuring eye movements while people are watching something is not enough, because we do not know what it means. So your design has to be sensitive enough to give you precisely the data you're looking for. And that particular data is, your hypothesis can be very specific to the type of eye movement, to what aspect of the eye movement you are aiming at, right? So you can be as specific as this, as a pupil measure within a certain duration, within like first 200 milliseconds of a certain activity. So how to design experiments that are that precise? So to take you through that process, I'm going to start with one of the papers from our lab. Seema Prasad is one of the PhD students who used to be here. Now she is a Humboldt postdoctoral fellow at TU Dresden. So this is one of her PhD papers. I'm just going to discuss not the details of the science of it, but just the hypothesis. How did we come up with a design? How is the design made in experiment builder? So at any point, if you have any questions, please raise your hand. We will circulate a mic. You can stop me then and there and ask. Because this is a very interactive session, this is not a talk. So feel free to ask whatever you wish. Alright, so to begin with, so the objective of this particular study was to see if a certain cue, like a visual cue, you all understand what a cue is, right? Something that gives you information about something else. So it in itself, that object in itself is not meaningful, but it's indicating something else. So that any object that can become an indicator for something else which is relevant to your task or a goal is a cue. Now how a certain types of cues influence where you look. A saccade is basically an eye movement which is a displacement from point A to point B. From the title that perception is attention research deals with a lot of small, very finite, very small cognitive events that are very difficult to tap into with manual reaction time experiments, right? So unlike decision making, which takes a lot of time and it's a long process, attention is a very quick process. So to understand such quick processes, it could be memory retention, for example, again happens over a period of time, which gives you more time. So your experiments are a little more flexible in some sense, that you have a large time window to measure your data, but in these specific experiments you have a very brief time window to measure data, which is why the experiments need to be designed with that precision. So within that time window, the participant should be using the function that you are assuming that they will be using attention in this case, and the eye movements are required to manifest that process are also happening during the experiment. Alright, so I will just go quickly to the design because the theory is not relevant to us right now. Alright. So is it clear to everybody? Is it not? Is it not clear to anybody? Can you raise your hand? Increase the is this better? So like I said, the idea here is to see if a certain cue influences your eye movement later on the task. So the task goes something like this. You see a series of screens, okay, on a computer, and the end goal is to make an eye movement to one of these four spheres. Okay, very simple. So what we are doing is trying to understand how and what particular sphere do people select to make an eye movement to, which is called a free choice Saccard task. Why is it a free choice? Because the choice indicates the existing bias that you may have with which you are making the choice. So to understand the underlying bias, you give people a free choice that you have four spheres, make a Saccard. Now, how we are interfering with their original bias by incorporating a cue beforehand, right? So if you look at this particular screen, first you see a fixation, so you establish that there is a starting point for eye movements. Because when you're looking at a screen, you might make an arbitrary eye movement to anywhere in the screen because there's no object on it, right? So even when you're measuring eye movements, you need a frame of reference, a starting point from which everybody is looking at. So even if, so what I'm trying to say is, when you're comparing latencies or the time it takes for you to reach a certain object on a screen can be different for different people if you do not give them a same starting point. Which is why you always have a fixation. It doesn't have to look like a plus, it can look like a circle, it can look like an object, it can look like anything. It doesn't also have to be at the center. Here it is at the center because the design is diagrammatically arranged around it. So it can be completely on the side depending on what your task is and what you want the starting point to be. Typically in designs you might see fixation coming at the center, which is a little confusing because you might think you might have to just put it there. There is no such rule. It depends completely on your design and your frame of reference. So, okay, so once you have a fixation, I will come to how you do it, but you have to design your experiment in such a way that it senses eye movement on that fixation. Okay, so there is a certain way to do it in experiment, but I will come to that. So once you have established that participants are looking at the fixation, then you can start with the task. So here the task has one screen which just places the circles. They are called placeholders because you know that is where you're supposed to look at eventually. It just gives you an idea of, okay, this is my visual field and this is the relevant visual field where I'm supposed to act at, where my task is associated. So once you've done that, then there is a cue, which is your manipulation in this task. So a ring brightens up one of these placeholders. It can be any one of these four, right? So this brightening happens very quickly for 16 milliseconds. The reason for that primarily is to make sure it's unconscious, that people do not see it. 16 milliseconds is a very short time for people to be aware of what they are seeing. But at the same time, because it's a bright ring and black and white is very contrasting, you will still feel like you have seen something. So that's the nature of this cue. It's an abrupt onset cue, so it has to come abruptly and make an impact but not get perceived that consciously. That's also besides the point. So you see a cue and then there is a duration given a variable duration for the queue to affect your existing bias. Because you want the queue to interfere with your existing plan, if you have a plan. You always have a plan. It doesn't have to come because of a strategic reason or a goal, but there is always some plan. You choose to make an eye movement somewhere, right? So to make sure that the queue gets enough time, or you have a variation of time, like it is there, like there are 3 SOAs, which is 3 specific durations for which this particular screen appears. So the screen can appear at any of these 3 durations, and it's randomly presented during the task. So the participant cannot guess or anticipate at what point of time the actual screen at which they have to make an eye movement appears. And then the final screen appears. So once the queue has interfered with the decision of where the eye movement has happened, and this is where the screen, you can't see it right now. So at this particular screen is where the response happens, and you get some 1000 milliseconds to make a saccade, which is more than enough time to make a saccade. And you get a feedback. It can be too slow or it can be wrong if you have not made the eye movement. So is the design clear? Yeah.
What is SOA?
SOA, I spell out the abbreviation, is stimulus onset asynchrony. What it means is the time between your actual stimulus onset, which is this. This is your stimulus because this is where you're supposed to respond to. Before the onset of the stimulus, how much time are you giving between the cue and the stimulus? So why there is a variable here is because if the cue, let's say you have perceived the cue and in 33 milliseconds you see your response screen. Now you make an eye movement. Versus you see the screen after 50 milliseconds or 300 milliseconds. The longer the time that you have, your goals influence more strongly. In theory, in the attention literature, the idea is that if you give it more time to come back to what your existing goal was, if something external is affecting your decision, the goals can override that decision. If you have enough time, so that how do you know how much time is enough time? So to come up with that number, we use SOAs that are of a certain range. So something very fast like 33, something a little in the middle like 50, and something slow like 100. Now, how are these exact numbers coming here depends on the literature that you are reviewing. Depends on your particular task. Depends on the type of goal, how strong it is, because here it's just about saccading. So 100 milliseconds is sufficient. It's slow enough because it doesn't take that kind of time to just decide to make a saccade. But if it was something like a discrimination, or like Amitash sir just showed some slides, like you have to make a judgment between 2 images, might take slightly longer, and it involves more than one saccade, of course. So, did I answer your question?
The stimulus and cue are coming in the same screen at the same time or a different time?
At different times. They come sequentially. So,
like all 5 are coming in like different time or in the same time?
So you see this line goes. So the sequence of events is fixation. Then you see all these 5 placeholders.
Okay, all these 5.
On that 5, this ring is superimposed. It's additional. Then the 5 continue. The ring is gone, which is your cue. You don't need the cue anymore once the 16 millisecond is passed, right? And then this comes. So this, as soon as the blue goes away, you know you have to respond. You see there's no blue here. That's a signal for you to act. Is that clear? Anything else? Okay. So if we are clear with the design, I will now start with experiment builder on how to make it. So can you just tell me if according to the hypothesis, the design makes sense to you? It doesn't make sense to anybody else? Can you tell me it doesn't make sense? It's clear? Okay. Okay, so this So this is what experiment builder looks like. So if all of you have experiment builder, can you go to File and click New and open this screen. So when you click New, it asks you to save a project. Give it some name. You can call it demo, whatever you would like to call it, and save it and come to this file, and then we'll move forward from that point. If you have any issues doing that, our volunteers are in the audience. You can just raise your hand and somebody will come and help you. So basically come to this screen. As soon as you save it, you will see an empty layout like this and a start in the center. Does everybody have it? Alright. So I will just explain the layout first. So there are, like you can see, there are several blocks here, and these blocks serve their own purpose. So this big block that you see here is the space where you drag icons and build your experiment like a flow chart. So why do I say like a flow chart? Is because these icons have their own boxes, and you join these boxes by dragging a line from one box to another box. Okay, and the flow of the line tells you the flow of the experiment. So it starts from one and ends at another, and wherever it ends, it creates an arrow. The arrow indicates that it flows from point A to point B. Okay, the direction of the arrow matters, because if you are dragging it the other way around, your design might not make sense. So you have to always drag the arrows from start to end. And how do you decide what the flow is? You reference to your design. Okay. So you have your design in mind. It has to look like this. So you follow the set of steps to make it look like this. Now I will explain the layout first. So you always save this file. This particular file type is called a source file. This source file will eventually convert into an application, which will be your experiment. So that application can run independent of the source file. You do not need to carry this particular file in every computer, in every system that you are using. As soon as you make it an application file, it runs on its own. So before you start your experiment or before you start collecting data, you have to make sure that whatever you design in Experiment Builder is complete. Because if you want to make any changes later in an experiment, once you have made it into an application file, you have to again deploy it. There's a function to do that, which makes it into another application file. So the previous experiments will not have the same manipulation that you're making freshly in the Experiment Builder file. So what I'm trying to say is, you have to, so there are also ways to test it several times, run and test it, like you run any other application before you make it a final application. So always do that before you're sure that you're ready to run an experiment. So now, if you, is my address, okay. So this particular window shows an overview. It just makes a diagrammatic map of what your experiment looks like. It's usually pretty useless. It's just for people who are very visual thinkers, who like to look at, look at modules in a very visual format, how they are placed and how it is. It just helps you visualize. There's no real utility of it, but like any designer that doesn't come from this field, you have your specific aims, whatever helps you visualize your experiment. So it is just one of those tools. So as you build things, they will make up like a map here in the overview. And if you want to ever go back or check what is where, that overview gives you a good picture. So you will see how it develops as we make a task right now. Then the structure does the same thing. This structure window does the exact same thing. It shows you a flow of the experiment, but with a tiny icon and a word. That word is a label that you give to that particular icon. Okay? So if you're dragging a screen and calling it fixation, you will see fixation, right? So why this particular type of view helps people who like to see more verbal sort of instructions. So this is my interpretation of why they are here, is I think it for different kinds of people who design, a different kind of tool will help them track their thought on where the experiment is going and what is lacking and what is missing and how are they fitting into each other. So this view helps those kind of people better. Now if you look at the lower window, there are three, it's a little blurred I'm guessing. Can you see this? What it is? Okay, so this is experiment, this is component, this is devices. Okay, so if you have your own computers, you can look at it. Okay, so when you say experiment, it shows the structure windows is a certain window. When you say component, it shows you all possible components that it can have. Now I'll explain what the components are. There are mainly 4 types of components. One is an action component. What an action component does is create a function, perform a function. It can be display something, produce a sound, play a video, or just recycle a certain display. Any kind of action that you might need to do during an experiment. These action components, which are basically displayed here on the top, as you can see in the top bar, all of these are action components. Okay, so these basically perform a function. Then there is another component called triggers. So what triggers do is navigate from one action component to another. Okay, so I'll give you an example. If your action component is, according to our design, first one is make a fixation screen, right? So now when you have to move from fixation screen to the second screen, which is this, how does that happen? The transition from one action component to another action component. It should happen with a certain contingency, which is time here, right? So fixation is displayed for 500 milliseconds. As soon as 500 milliseconds are over, the next screen should take over, right? So the incorporation of 500 milliseconds relies on triggers. Triggers can be of several types. For this particular example, a trigger can be a time trigger, which is a duration, which you can see here. When you see the symbol, it looks like a clock. Okay. The good thing about Experiment Builder is, as you hover on these icons, it gives you a tiny dialog box which has a description over it. So the description on this says timer trigger triggers when the specified amount of time has passed. What this means is, as soon as you select this trigger and you enter a number in it, let's say 50, it means that after 15 milliseconds has passed, you have to also be careful about the units. This time is being measured in milliseconds. Okay? So after 50 milliseconds has passed, this trigger is connected to whatever action later will get executed. So the order in which you organize your actions and your triggers matter. So I will do a quick so let's all do this. Can you see this icon here? This particular icon. So what this icon does here is it creates a new window in which more actions can be built. What this new window does is basically it creates a layer within your experiment. Okay. Okay. So why do you need this? First of all, you would have imagined that on this one piece of paper, you could just drag everything, make your entire file, and it would be done. But Experiment Builder doesn't work that way. It works in layers. Like I said, you have to sequentially make your experiment. Okay. So the first layer usually is just the start. So as soon as that first layer just gets run, imagine a code being run. Let's say, imagine a code is placed here. Okay. So the first thing that you run is start. The computer gets a signal, okay, start the experiment. Then it will come to the second layer. The second layer is this. This is how you create a second layer. You just drag this icon down and drop it here on the white. We do not need 2 of them, so I'm going to delete it. Okay. So we just need 1. So we will go one by one. Okay. So you just connect. Did you see what I did here? I just dragged a line from start and connected it to this big box. So now you can see there's an arrow which points from the start to here. What this indicates is that this comes first and this comes later.
How do you get the arrow?
You just click on the start and drag it. Drag your cursor across. Is it generating an arrow?
Yeah. Okay.
Yeah. Everybody understood how we did this? Alright. So now how do we get, now the next piece of code has to be within the layer, right, like I explained. You just double click on it and you enter a new layer. Now you see it's a completely new, right? So now we are in this frame and on top you would see that a new layer has formed. You can see first one says experiment, second one says sequence. So we are inside sequence now. Now we will build a little more code. Okay. So what happens after you start an experiment? You might all know what is the next thing that you see.
The fixation.
Instructions.
Okay.
So the participant has to know what they have to do in the task, right? So the second layer that you usually build is your instruction screen. But if you're doing if you're doing an eye tracking study, you need to build a calibration screen before you start the instruction. So if you can recall the earlier presentations, there you see a dot on the screen, you have to look at the dot moves across the screen, you keep looking at those dots in that order, and what an eye tracker does is records your eye movement with reference to the dot. If it aligns with the dot, which means you are looking exactly where you expected to look and the calibration is successful. If you are looking far away from the dot, then you have to redo the entire process and align your camera and make a few adjustments so that your eye movement aligns with the dot. Which means, and once you have done with that, which means the calibration is successful. So every eye tracker has its own type of calibration. Depends on the company, depends on the software, what interface they are using. But the idea is pretty much the same. You have to basically map where you are looking and where you are supposed to look by an indicator. It can be a dot, it can be anything. So to do that in SR, there is already a node like this. It's called EL camera setup. Okay. So you can connect start to the camera setup. I know this looks ugly. SR has done this to make our lives better for those ones who are not very organized. So you just click on it and it organizes everything. This is beside this is the icon. Arrange layout. Okay. So click on arrange layout, everything comes back to the picture. Okay. So back to the camera setup. Like I said, in this particular interface, you do not have to code for you to calibrate or do anything. This particular icon, as soon as you drag and connect, aligns the code necessary for calibrating with your program. So now as soon as the start command is sent to the iLink, it will also incorporate the calibration code. So it will run the calibration on its own. You don't have to do anything about it. What you have to do later is to check what happens after the calibration ends, right? So you have to give, for example, participants a small idea that calibration is about to come. So if you want to do that, instead of having this first, you would want to have a screen which says, okay, now the calibration screen is going to come, please look at the dot, keep looking at it until it's done. So you would include a display screen before you do that. Okay. So now what I did is I connected start to the display screen and then the display screen to the calibration node. So how to, now because you've connected a display screen, which means you can show something on the screen before the calibration happens. Now how to add the text or the instruction that you want to give the participant. You double click on the display screen. Okay. So as you double click, something pops up. A blank space pops up. This blank space represents the screen that you have just added. It's blank because we have not added anything here. Now there are a range of things that you can add on the screen, right? We just discussed that we need to inform the participant that the calibration is going to come. So you want to add text. So as you can see on the top, whatever can be added here exists on the top. There is an insert image option, there is an insert video option, there is a one word text option, and there is a paragraph option. There are also shapes. It depends on what you want to use. You have any questions? Okay, so you just click on one of these icons, whatever you need, and click on the screen. So I clicked on the paragraph icon and clicked on the screen. So a paragraph dialog box opened up. Okay. So now I will just write.
Back and say what
Do it again.
exactly did you click?
Okay. So you have the screen, right? You have added the computer screen. You double click on it, and then something pops up. A blank area pops up, right? On that blank area, you can add whatever you wish to add: images, text, videos, audio files, anything that you wish to add. You can use also audio instructions if you're doing experiments with a certain special population that cannot read. Like for example, we are doing experiments with illiterate population. They can't read what we are showing, so we use audio messages for calibration, for instructions, for everything. Okay, so I wrote down something using this dialog box. Okay, how do you incorporate text? Is you go on that icon, click on that icon, and then click on the blank part. Then it pops up. Then you can write whatever you wish to write. So now you can see it's a different dialog box than earlier. Because I clicked on it again, you can have multiple text on the screen. But if you're using a paragraph, it doesn't make sense to have multiple dialog boxes, so you just might have one. And you can add whatever you wish to add on it. So now they're overlapping. Can you see the earlier dialog box and the later dialog box? Because the locations are different. You can use that if you have instructions at different parts. If they mean something, then you can have multiple dialog boxes and position them accordingly. And if you want to edit something existing, then just double click on that. So when I double click on this, it opens the existing dialog box. Now you are free to edit it again. And editing this is just like WordPad. You just write down, there are a bunch of orientations on the top, center, right, left, and you can change your font and however you want it to fit the screen. So I will not get too much into this because these aspects of the Design Experiment Builder you can explore on your own. Okay, so now assuming that on the screen we have told our participants that a calibration is going to happen, please follow the instructions accordingly and just keep looking at the dot. Then you have connected the calibration node to it. Now the calibration is taking place. What would you want next? You want the participant to now agree to start the task, right? So you would want another instruction screen to show that, because obviously you are not going to speak to your participant during your experiment. You would want this to run flawlessly without any human interference. It's very important. So you would incorporate another screen, and you want the participant to consent that okay, now I am ready to start the experiment. So you will use a trigger. You can use a time trigger that, let's say it takes 200 milliseconds to read whatever, it takes longer, but let's say it takes 500 milliseconds to read whatever instruction you have given. You can give a time trigger. Okay, after 500 milliseconds, it goes to the instruction screen. Or it is advisable to use a keyboard or a mouse. So participant can click that they are ready. So when you make this display screen, the display screen 2, in the content you can particularly mention when you're ready to start the task, press spacebar on the keyboard. Okay. So when they do that, they can do that because the trigger is connected here. I just dragged the spacebar, brought it here and connected these two. So now participants are allowed to press buttons on this keyboard, whatever keyboard is connected to your computer. Is that clear? I see some confused faces. Is there a confusion here? You can ask me whatever you think is confusing. Do you have any questions? Alright. So as you connect a keyboard, which means, so now also focus on the line. I have connected two things. I have connected a timer and I have connected a keyboard. Why there are two things is because if let's say they do not press a button, if they forget about it, it might be hung here for a very long time. You don't want that to happen, because some you might also want your participants to be a little more vigilant about your task. You don't want them to take however long they want to take. So just add a small timer and give it a certain value. So it also runs for a definite period of time. So when you connect two action objects to one action object, both are active simultaneously with this particular action object. Now, if you want one to get over sooner, you can define it. So as you click on these action objects, you can see in this dialog box a properties dialog box opens up. Now this properties dialog box is specific to what you have clicked on, the action object that you have clicked on. So because I clicked on timer, properties of the timer shows here. Okay. So if you just look at these properties, what does it say? It says a label, like I said, which is displayed on the structure here. You see the label, which is timer. You can change it. You can make it anything, like a instruction screen. Right? So now the label is changed to instruction screen. It's very simple. Then you give it a message. Message is useful while extracting data from the data viewer. We will come to that in tomorrow's demo. So usually what you try to do is give a meaningful message. What I mean by meaningful messages, you should know you shouldn't give it as screen 1, screen 2, because when you come back to it 10 years later, you will not remember what screen 1 and screen 2 is. You have to give it a certain name. That is it fixation, is it instruction, is it stimuli, whatever it is. So give it a meaningful label. These labels are very useful. Messages, I'm sorry. Give it a meaningful message. These messages are useful when you're extracting data based on these messages. So they should be unique and they should be meaningful. So once you are done with your messages, you look at the duration of your timer, because this is a time action. So right now it's set at 4000 milliseconds, which is a very long time for participants to just understand that now they need to start the task. So you might just keep it at 100 milliseconds, which is enough time. Again, how much duration do you want to use for a certain timer or a certain display totally depends on your hypothesis. It has nothing to do with the design software. It depends on what you want it to be. So you can just edit it here. These functions basically just work as typing. You erase what exists and you type down what you want. So it's 100 and the unit is milliseconds. Alright. So once you do that, which means the screen will be shown for 100 milliseconds or until something is pressed on this keyboard. Okay. So when I click on the keyboard, sorry. When I click on the keyboard's properties show up. Now there are some commonalities like the label and the message.
So I can't get rid of the arrow that's coming actually. Just the arrow keeps on hopping again.
If you do not click on the icon that you want to connect to, the arrow keeps confusing where it needs to go. You have to click it clearly from point A to point B. Okay, so when you click on the keyboard, there are two common properties, label and message. There is a different property which says keys. So if you want to specify what key they have to press, then you specify it here. Okay, so how do you do that? You click on the left hand side. You click on this bar where you see any. You click and this dialog box opens. Did you get to know how the dialog box opens? You just click on any. Beside keys on the right hand side, in value, just click here. When you click here, a dialog box opens. And that shows all possible keys on a keyboard. Okay, so you can just select select something. I am selecting space here. Can you see it's selected? I select it and I close the dialog box. Once I've closed the dialog box, it shows up here. Now this says space, it doesn't say any. Any means any key could have been pressed and the trigger would have worked. But I want my participants to be a little more vigilant. They should read it and actually press based on the instruction. So when they read press space, they will press space. And no other button will work on the keyboard. All the other buttons will be blocked on the keyboard because you have coded it so.
Can we have multiple response keys?
Yes. So how do you do that? Is you click on the dialog box again, click Control and select multiple options, and you just close it again. So now you see there are a range of options here. All those keys will function. So keyboard and the mouse and this another keypad, I-link button trigger and the Cedrus input trigger works pretty much the same way. It is a input button. That button can be on a keyboard, it can be on the Cedrus pad. It's nothing but a response pad with minimal keys. A lot of experimenters use the Cedrus pads for one reason, which is that the interface between the press and the button getting recorded in the computer is a little faster, 2 to 3 milliseconds faster. Doesn't make a lot of difference in many studies. Makes a lot of difference in some studies. Depends on your experiments and what kind of experiments you're doing. Or you can just use your keyboard and have any particular key, or you can use a mouse. So the difference between these will be that the keys will be different. For a mouse you have a 1, 2 and 3 based on a 3-click mouse or a 2-click mouse, or a scroll forward and a scroll backward. So you have 4 keys. Forward is 4 and 5 is backward. So instead of enter, clear and space, you will see 1, 2, 3, 4. So you can select what do you want, a first left click, right click or whatever.
Instead of using dialog box, can we type it directly?
You can type it directly. You have to make sure that it's case sensitive. So you use the right case. So it's C is capital and LEAR. So you have to write it clearly. It does indicate that. Also when you start typing, it will also pop up. So you don't have to really type. It will just pop up and you can select it. Yeah.
With the mouse, how you said 1, 2, 3, 4, 5, it's like predefined. So you know how certain mouse keys, they have like this thing where if you're like a left-handed person, then you set the conventional right click to the left click.
Yeah.
So then does that numbering change in those cases?
Yes.
It changes. So then the 1, 2, 3, 4 will be for the right, left, and then so on and so forth.
So what you can do is, if handedness is a factor for you, then you make sure that you know what your 1 and 2 and 3 and 4 is for your particular mouse.
Okay.
Right. So when you build a data source, which you will have to build here, I will come to that quickly. In your data source, you can specify the keys.
Okay.
That 1 means this, 2 means this, 3 means this. So every time once you have the data based on your participants, you can compare all this. I know right now it might not be very clear, but when we're showing the experiment, I'll make it clear. Okay, so this is what we need in the second layer, and we're done with it. Right? Now we have to go another layer in, because once we are done with the calibration, you have to do the experiments instruction, right? So you can also see that 2 nodes can connect to one dialog box. Okay. So now we are ready to go inside the next layer. Is everybody with me or is anybody lost?
I can't find the arrange button.
Arrange layout is what you need to go to. One second. Got it?
Yeah.
Okay.
Can you please show again what is after arrange layout?
You see this icon here? You just drag the icon down and then connect the keyboard and the timer to that big block.
Can you show it again?
Right? So you have to make sure whenever you are leaving any end, you have a loop to go back to at least. What I mean by that, I'll come to later. So are we all here Is anybody confused or stuck anywhere? Maldika, can you just help them out? Okay, so now we are ready for the instruction screen, and now all of you know how to do that, I'm guessing. So you just drag the display screen here,
How do I zoom?
double click on the display screen, drag click on a paragraph, click on the screen, a paragraph pops up, and now you write instruction. So you've written your instructions, and that's it. Now the participant just needs to read the instruction and start the experiment, right? So again, I am connecting a keyboard to the display screen so that as soon as the instructions are read and they are ready, they will press the key and start the task.
Label the sequences.
Yeah.
So what will happen here is that from this sequence you were here, right? You added these two, then you went inside of this sequence. Start already playing up. All I did over here is
Add the display.
so you have to do that inside. Add the display screen.
Okay, so I have labeled this particular keyboard instruction key, so that there are other keyboards. You can also see the previous keyboards from the previous sequences. They also have the name keyboard. You should not get confused by that. So you have to know, like I said, the labels have to be meaningful, right? So okay, so now once we have this, we will start actually building the task. Okay, so you create another layer, and this layering is important. You might think it seems very unnecessary and too much of a hassle. This layering is important so that your program doesn't confuse between task A to task B. You have to just basically keep it clean, so your code is very clean at the backdrop. You're not writing a code here, but as you are dragging things, the code is being written at the background in the software. You don't want that to be very messy, which is why you create these layers. So now we are moving into the main experiment design. There is another additional layer where, now this is also the most important layer. Here you add your breaks. Okay, you might have to give your participant a set of breaks when they are doing a task. That is not why this layer is important. It is important because you add your data source in this layer. So what is a data source? For you to run any task, there are a range of conditions. Like in this particular task. Okay, so as soon as it shuts back. Okay, so in this particular task, what is your manipulation? A queue is coming in one of these 4 locations, right? So the queue is same in properties throughout wherever it's coming. So you don't have to do anything about that. But it has to come at 4 locations, and that has to be random, completely random. You can't, the participant shouldn't be able to predict where it's going to come, right? And you don't want to now manually do it. Like you don't want to show it in some and then show it in some again. So to do that, what you would do is have a column in a data source which tells the program where the location of the sphere should be. So you make it an independent object. You just make a white circle. Just make an image of it in Paint. You can do anything. You can use whatever software that you want to use. Just make an image of a white sphere with a transparent background. That image now you are using in this design and displacing it from one of these 4 locations. Okay. So for that you need one column in data source saying location.
So data source can be any CSV file, right?
Data source can be any CSV file, preferably a text file. So what you can do, the easiest way to do it is do it in Excel or any sort of a tabular format and export it in text when you are exporting it here. So you can make it in your comfort, whatever file that you feel comfortable with. When you are incorporating it in here, there is an export option. You just convert it in text while at that time.
And for the position of the queue where that is going to come.
Yeah.
So are we going to use coordinates or something to locate
Yes.
that?
Yes. I will show that once we are doing the data source. So I am just dragging another here, another block here again, because in that block we will put the exact design. Right now we are making the data source. So between these two should come your break sequences. That is just like a screen and a timer. Okay, a screen says you now have a break, and it stays for, if you give a 2-minute break, then it stays for 200 milliseconds, 2000 milliseconds, right? That's it. So I'm not going to make it here because you all know how to do that.
Can we draw the circle directly here?
Sorry?
Drawing the circle directly here, not taking the image as
You can draw the circle directly here as well. Images are just easier because I think drawing takes longer. Images are already preloaded in the design. As soon as, no, so not right now. When you're running an
One
application,
circle and then copy and paste it.
you can't copy and paste here. You can just draw on a particular screen. I will show you how to do that. I will show you both options. Okay, so in this, how to now add a data source. Can you see here, there is a dialog box which says data source in this layer. Do you all see it? Do you all see the data source here? Is anybody who does not see a data source there? दिख नहीं रहा है? Okay. Everybody there? Can you see the data source? You all see the data source? Okay. So you click on the data source, and this window opens up. How much time do we have?
1 hour sleep.
Another 15 minutes. Okay. So when you click on the data source, does this file open?
Yeah.
Okay. So on this dialog box, you can see on the top there is a add column, add row, and import data option. Okay. So if you want to manually do it, like if you have very little things in a data source, for example, just location. So you can, if it is just one column, you can just add a column. Click on add column. It opens a small dialog box like this. You give it a name. For example, here we need location, right? So you give it a name, location. Now you have to specify the type of the column. So somebody asked, like, do you give coordinates for the location? This is where it comes into the picture. So when you specify what is this data, so what is this particular column in your experiment, it is a string, which means it just has to read it like a string. It can be anything to you. 45 minutes? Yeah. So it can just read it like a string. For example, if you have to display a text and you are displaying multiple text for the experiment, the program doesn't have to interpret the text in any other way or place it somewhere. In that column, you are just giving a set of alphabets. So it will just read it like a set of alphabets. Okay. But here you want to give it a location. So you will not choose a string, you will not choose a number or a color, you will choose a point. So a point is a Cartesian x, y point. Okay. So when you're writing a point, you have to also insert the data in such a way that it's x, y. Now the x can be, and what is a certain x and a y on the screen? How do you define that?
We need to know
It
the
depends.
extremes of the screen.
Yes, it depends on the resolution of your screen. So if your resolution is, let's say, 1280 on the certain x-axis and 1244 on the y-axis, you can check your particular screen's resolution. If you see that, so then this point in your screen, this top left point will be 0, 0. Okay, x increases this way, y increases this way. Is this clear? So what I mean is, if that point is 0, 0, what would be the center of the screen? You just divide your x into half,
Half and y into half.
y into half, right? So that point will be your
So
center.
do we have to put a negative value to y since it's down?
There is no negative value because 0, 0 is the start, and it ends in a positive, and this ends in a positive.
Okay, so this is also positive.
Yes. Everything is positive on the screen. Okay? So if it goes down, y is positive. If it goes towards the right, x is positive. Okay? So the center of your screen will be half of your x and y resolution. Then if you have to place something here, you just add those many pixels and place something here. And again, add those many pixels and place something here. So you should understand how a Cartesian coordinate works. You can just, the easiest thing to do is draw it up on a certain graph. There are several these graph kind of softwares where just a Cartesian coordinate. Place things on that, you get an idea, then make it on your I will also show you those softwares later in the tasks. Okay, so you do that, and if you have a default value, if it's like a ordinal variable or a numeric variable that goes from some point to some b, then you can start a default value. If you do not have a default value, leave it blank, click OK. So now it creates the column here. Now it says location, it tells you what type of category that variable belongs to, which is point. Yeah.
So now if you're running the experiment on another screen, which is not the screen we designed, and obviously, like you said, it's already made into an application. So now, suppose the resolutions don't match. So there's no way that this automatically will adjust to the resolution of the new screen.
Yeah.
Is it?
Yeah. So it will adjust to the resolution of a bigger screen, but not to a smaller screen.
Okay.
Okay. So it can go, if you have designed for 1280, it can work on 1366,
Okay.
but it will not work on high. So what it does to adapt to a smaller resolution is it will just shrink the window, and the sides will become black. You don't want that. So what you usually do is, if you're anticipating to collect data on another PC and designing on a different PC, design it keeping the data collection PC in mind. Whatever resolution you will be collecting on, you have to use those specifications. The drawback of that method is that you will not be able to test it while you're designing simultaneously. So what you have to do is, you've designed, you deploy it. I'll tell you what it is. Deploying is just a button to make it an application. Once you deploy it, do a shift to another computer and run it there. You have to keep doing this back and forth if you're changing resolutions. Okay.
How
So
do
now,
we decide the size of the image if we are importing the image?
yeah. So okay. So when you're doing anything, I will speak from the point of view of visual cognition. Right? You're measuring eye movements. Your participant is sitting at a certain distance. When they are viewing an image, a visual angle will be cast at the image, right? So the visual angle has to be calculated based on your design. How much do you want the visual angle to be? Based on that, you will design a size. Okay. So SR Research on their website has a visual angle calculator. You can all check it if you want to check it right now. You have internet. Just search SR Research visual angle calculator. When you open that, you will see a range of options on where the objects are placed on a screen. Is it placed in the center? Is it centered in the center? Is it placed on the sides or on the quadrants, in the center of the quadrants? Wherever it is placed, there is a formula to calculate visual angle. So you can just specify the dimensions of your screen, distance of the screen from the participant, and what else? Resolution. The resolution of your screen. So when you specify these three parameters and you enter you can check the visual angle for any object. If you want it to be, let's say 50 pixels, you can check what visual angle it is. Then you can go back to your reference papers, whatever you're using to design your experiments, and see if it matches. If it doesn't match, you can tweak it a little, make it smaller or larger. Does this make sense? You can check the visual angle calculator today. If you have any questions, come back to us tomorrow, we will help you out. Okay, so now we have added a column, we need to add multiple rows. So in this data source, multiple rows are your number of trials. Okay, every row is one trial. What do I mean by one trial? Is one run of this task. Yeah, one entire run. Fixation, queue, response. That's one trial. Okay. So if you want, let's say, 50 runs of it, you have 50 trials. And what decides the number of trials? Again, your particular task. You have to go back to your literature. So I am adding 50 rows to this. So what it did here is created 50 rows. Okay. So now you can, like I said, make something in Excel, import here. You don't have to keep filling this. But if this is all that you are making and the task is very simple, you don't have any other variables, you might as well just fill it here. It's not that easy. So as you enter a location here, you have to open brackets. Okay, so let's say 560 comma 800. I'm just giving an arbitrary number. Okay, so this is how you write your positions. If you open a bracket, write x comma another value y and then close the bracket. And then you just follow the same system here. Make sure you don't add spaces in the middle. Don't add anything else that can be confusing. So because the notation is pretty standard for a point. Even if you import from Excel, it will automatically convert, SR will automatically convert it to its own format. So you don't have to do
We
it.
have to just do it in a text format.
No, you do it in Excel,
Yeah.
you download the file in text.
In text, right?
Yeah. So is this clear? So I will just add another type of column for the sake of it. Let's say the ring had color. Let's say the ring has multiple colors. Okay. So let's call it color, and you select the type color. Okay. Once you select the type color, now instead of 2 values, you have to give 3 values, which is RGB. So you can give it 153, 153, 153. This is a certain grade that I use for my experiments, so I've learned it very much. And that's it. Okay. So this now what will happen is if I link any object to this column, I'll tell you how to do that. When you link to this column, the color of that object will become this particular RGB.
So whenever I'm putting 153, there's a 255 coming at the end, right? After 150.
Yeah, so sometimes there's a RGB and alpha, doesn't matter, because it's constant throughout, so it will always be 255. You just write your RGB. Like I'm not sure why though. I don't understand color theory that well. But okay, so this is it. This is how you add your variables. If you want to import it from outside, I'll just show you that. So you see this import data option. Okay, click on import data. You click on these dots wherever your file is. Click on that particular file. Click OK. And here you have to specify the delimiters of that file. For a text file, what does delimiter mean? What separates one column from another column? Okay. So for text, it is always tabs, which is by default, which is space, this space between these two columns. It's always ticked. For other type of files, there are different delimiters. So you, based on what type of file you are importing, you just have to click those delimiters. So the software understands what type of file you are using. If you're using an Excel and you've kept tab, and when you're making these columns, you have basically, let's say you have named one column location of target. So it might take location as one column, off as one column, and target as one column. You don't want that. So if you want to have like a multiple word name, use underscore to connect them. So you don't have any tabs.
Also, for the location, for this particular design, we have 4 locations where
Yeah.
the Q can appear. So, since we ideally want those to occur randomly, so we can put the 4 locations and
Then
we
repeat
obviously
them.
drag it and then have
Yeah.
it.
Yeah.
So, what determines the randomness of the?
Yeah, so I'm coming to that. So as soon as you fit your data, you can make, so let's say you have, you write all the individual iterations of your data, okay? Yeah, here there are 4 rows, 1, 2, 3, 4, wherever it comes. Now you can go to randomization setting. Okay, do you see this? Do you see this icon, randomization setting? You just click on that, you click on the column that you want to randomize. It can be anything, it allows you anything. Okay. The good part about this is, if you write a code for this, it becomes very confusing when you're randomizing things. It might randomize one, block the other, block one and randomize the other, block both, you don't know what is happening. So that gets a little tricky. Here you just have to do one tick, it randomizes it and it blocks on its own, it makes structure, you don't have to do anything now. So let's say you click location, in the randomization just tick randomize. That's it. Then location will be randomized. No two same values, consecutive rows will appear simultaneously in an experiment. That's what it means. Like I said, your row is your trial, so basically same types of trials will not appear simultaneously. So when you write when you make a data source, make sure you are writing it that way. Everything simultaneously, keep it one after another. So when you randomize, it just picks whatever it wants to pick. It doesn't go one by one. Then I select color also and I click randomize. So both these variables are now randomized. The random color will come, random location will come. Now there's an interesting feature here. It's called it's called run length control. Okay. So what run length control ensures is that if, let's say, even with the randomization, two types of trials are very similar with all same values. If they happen one after another, how many such trials can occur one after another? So this question got asked at some point, I don't remember, in our lab, that a certain type of trial is coming one after another. Right? So you don't want the participant to learn a behavior while they are doing your task, which is completely unintentional to you. So what you do is you control that, okay, only two trials can come simultaneously, because if there are three trials, it's enough to make you feel like it's a pattern. If it's one trial, of course it's completely random. But if it's two, then it might feel like, oh, this is a happenstance. You don't learn anything from it, as long as it's not three. So 2 is typically a number that people use. That more than 2 similar trials will not reappear together. So you just click on run length control and you just specify the number here. You can change it. You can make it 5, 6, 7, whatever, depending on your design. What you think is the degree at which people learn these things. And that's it. When you're done with all these settings, you can click OK. Okay.
Not being able to click on that maximum run length.
Sorry?
The maximum run length, which is
because this is a demo version, it doesn't allow you to change. In a licensed version, you can make a lot of changes. So the demo version has some limited features. Not at very complicated levels, but at these little things, it won't make much of a difference. Again, so what I forgot to explain all of you is here, you see there's something called a blocking level. Okay. So what I have done is also block location. Matlab, a certain type of locations will come together. Within those trials, it will get randomized. If I have one value per location, those one will always come together. Within that, it will get randomized. If I want to just randomize it without blocking, I don't need to add it here. I will just click here, enable trial randomization. It will just randomize it on its own. But if you want a group of trials to come together based on a certain variable, like red colored cues will always come together. They can come anywhere, but red colored will always come together. So I will block color. Okay. Add color to this list and nothing else. And click within that, click randomize. And not add length, not add location to it. So what will happen here in this design is it can come at any 4 locations, but a certain color will always come together. Oh, is this clear until this point? Any questions?
Can we also create blocks of certain trials in this? Like if I have to run 10 trials,
Yeah.
and then I want to repeat those
10 trials.
in a similar pattern.
Yeah.
Can we create
So you have to categorize those 10, differentiate those 10 trials from the other trials based on some variable. Okay, if you, let's say you want to do a practice, okay, and you have a column which says practice for those 10 trials, the corresponding value of that column for other trials says something else. So what you can do is split the data into those categories. At the bottom, do you see something called splitting column? So the splitting column is used for this function. If you have different types of trials that you don't want to run together, you want to just keep running those split columns, and then when you're done with it, you have to run the other split columns. So then you can just click one variable. Whatever is your variable, experiment, practice, whatever variable that you want to create, which will split it, you just click that and select it, click OK. So when you run the experiment, it will show you two options based on how many factors that variable has. Let's say if location has 4 categories, right, left, top, down. Okay, if there are 4 these values in location, when I run the experiment, it will show me a drop down list, right, left, top, down. What do you want to select? What it has basically done is split this entire design into 4 parts based on that variable that you have selected. So it split the data. So I can run right, 10 trials are over. I have to again start the experiment. Then I can again run right. You know what I'm saying? So if you want to repeat those trials, you can just do it that way. You can create whatever meaningful variable you want to create.
Then for practice trials, we'll have to add another column also.
Yeah, you can just make a column called type. So we can have practice and you can have experiment, you can have whatever you want. So is this clear? We'll move forward. We have half an hour. Okay, so the major part of the design is over. Now is just dragging and making those images that you see here. Okay. Yeah, this is your main experiment. But this is the less tedious part of the design because the more tedious part is the data source is already done. Okay. So now it's just visuals basically. So there is one screen with one object, another screen with a bunch of objects, another screen, another screen, another screen. That's it. And in the end, you have to make a card. So the trigger here will be a card trigger, not a button press, unlike before. That we will just look at. Okay, so let's assume we added a bunch of display screens with all the images that you want to add. Now, okay, right. So on this, I'm assuming there are some default images here. There is no vision level. I will just quickly explain how to add images and audio because it is different than just writing text. So if you want to add any image or a video or an audio which does not get created simultaneously with the design, you have to import it. Okay. So when you save this file, like in the beginning you save this file in a project, right? Then it creates a bunch of folders. One of those folders is library. Okay. In the library folder, whatever you want to import in your design, you just add. Library again has categories. It has category of images, audio, video, movement pattern. So depending on the type of the thing that you want to import, in images folder, put the images. In audio folder, put the audio. You don't have to do anything. You just have to put them in the folders. As soon as they are in the folders, you can import him here when you open your design. So you click, this is the image icon, click on the image icon, click here, unlike this dialog box because right now there are no loaded images. But if there were loaded images, you would see a drop down list with the names of the images that are there in the folder. You can just select what image you want and it will pop up on the screen. Then you can place it wherever you want to place it. I can add an image from somewhere. We have absolutely no images in this. Okay. It has no default image. This is SR's name. Alright, so is the image part clear? Similarly, there is a sound tab, right? That is for importing. So what I'm opening is the library. You see this column, this icon which looks like a building, that icon is your library. Whatever goes in the library folder shows up here. Okay. So if you don't see anything here but you see in the folder, it could possibly indicate that your file in the folder is corrupt, which is why it's not showing. So if anything can be loaded and can be used in the design, will show up here. Whether you want to use it or not does not matter. If you don't want to use it, let it be in the folder. It does not affect your experiment.
Do we have a particular format for the image or the video? Like
It can be anything. It can be anything. There are of course a more preferable formats like JPG, PNG is more preferable for images. Audio can be anything. .wav is works best. The other formats can get corrupted. You can't really tell. Also, if you are good at understanding the reasons of corruptions of files, you might be able to crack it. But if you don't want to that hassle, just use .wav. Okay, so you use your sound. Interest area pattern is specific to designs where objects are moving on the screen. So you want to specify in what order will they be moving on the screen. Like in a multiple object tracking experiment, for example. There's a ball that dances around the screen, then it freezes. After a delay, you see another image. You have to guess whether the image was the same as previous image or not. So if you want to specify what pattern in which that object is moving, you can just use an interest area set, which is just a collection of locations and the trajectory in a series. It goes from point A, point B, point C, like that. So, and then there's the video. Video is pretty much the same thing. You just import it a file, whatever exists, and just move it here. Okay, so that's about importing. So once you have your stimuli on the display screen, whatever is your experiment stimuli, which is this design right now, then you want an eye movement to go to one of the 4 locations, right? So on your screen you make these 4 locations. You make these 4 spheres. Okay. Now if you have to detect eye movement going from the center, which is the fixation, the blue square, blue circle. Of course the blue circle is not staying when you're making the eye movement. So the participants don't know, but for you to know that the eye movement started from there, you make an invisible area in that region. Okay. How to make an invisible area? You click on your display screen and you see these icons here. They are, it says interest area region. So the first one is rectangular interest area region, second is circular interest area region, and the third is freeform. Okay. So because our object is circular, we will use a circular interest area region. I click on this, I drag and make one interest area region here. Okay. The size and the location, everything can be changed. So what this interest area region is doing, it will not be visible to your participant when they're doing the task. It is visible to you. It's a frame of reference for you to understand within this region when eye movement occurs, it means something. It means fixation. When eye movements cross this, it means they're making a staccato somewhere else. Where they are making, you have to place interest areas at those locations also. So in this particular experiment, you need 5 interest areas. 1 in the center and 4 at the 4 potential target locations. Which means this is a free staccato, they can look anywhere. So need to have interest area at all locations. So, and like anything else, so I am just roughly placing it in these locations. Okay. I have not specified any location, but of course you can't do that in an experiment. I will just show you from one object. If you click on this object, again look at the properties. Typical properties label. Okay. There is an ID for an interest area, which is a number. It has to be unique for every interest area. If it means something, let's say fixation is 0, top left is 1, top right is 2, then there is 3 and there is 4. So it has to be unique for every interest area, you don't overlap it. It is just like an identity. So when you're extracting data, there is a column in the data viewer, which you will be told in the next session, which tells you where the eye movement occurred, at what location on the screen. So if it is within an interest area, it will give you the ID of that interest area. That okay, it occurred at ID 3. So for you to know, you know what ID 3 means. If it's multiple things, then you will not know what 3 was. It means multiple things. I could be in multiple locations. But because you want to interpret very precisely, you give the unique ID. Of course, if you forget, you can go back to your design and see what 3 was. So you don't have to remember it, but you have to make sure you're making it unique when you're designing. So you have a label, you have an ID, then you have a location, x, y. So this location you have to specify. Again, you can use the SR research visual angle calculator that can also give you the location or where it has to be. Based on how far it is from the center, what is the visual angle from the center that it is casting. Or based on all those calculations, you can decide what the location is going to be. Just write it down here, x, y, and the width and the height, which is the size of your interest area. Now there is no specific rule on what should be the size of an interest area. Okay. It can be as big as you want, it can be as small and as constricted around your object as you can be. So if your object itself is a very big object, then interest area can be very closely surrounding it, because you know eye movements, you know how finite eye movements are. If the object is very small, eye movement might just be on the edge of the object and you don't know what it means, right? So what you can do is you can create a large interest area. So you don't want to miss out on eye movement that is just outside your object. People might just be seeing it. Eye movement is landed a little incorrectly, because our eye movements, however we want to think about them, are not that precise. We might perceive without looking exactly at the object, which we do a lot of times because covert attention is also at place, right? So we are seeing without moving our eyes. So to avoid that, have interest areas larger than your original objects always. How large you want depends on your design. So it should not overlap with other interest areas. That is another critical factor, because if it does overlap, when you are measuring the eye movements and extracting the data, it will give you both IDs. That, oh, eye movement belong to both these IDs, and then you don't know what, right? So you have to also make that separation. A thumb rule that I usually follow is make them as big as possible until they touch. This applies when, so because I study visual search, searching is a very arbitrary behavior, very random, so eye movements can be spread around everywhere. You can't really predict how focused they are going to be, which is why you make it big. But if you're doing a Posner queuing task, or if you're doing a saccading task like this one, then it's a finite dot that you have to look for. You're not specifically searching for the dot. So your interest areas can be very precise around that object. Again, the participant cannot see what the interest area is, so it does not matter what shape you use. If it's a circle object, you use a square interest area, it does not matter. The only difference is it doesn't fit correctly. So you would want to usually technically match the shape of your object, if you can. Otherwise, just use a circle for everything, because it is, you know, like more uniform everywhere in all axes. How much time do we have?
15 minutes.
Okay, so I will quickly come to the eye movement triggers. It is the important part here. Now, when you have added objects on your screen, you have your interest areas. Now you want to detect a saccade to that interest area. If it lands within the interest area, you have to mark it correct. If it does not land in the interest area, you want to mark it incorrect. Here in this particular design, there is no such thing as incorrect. They can look at any 4 objects. It's a free choice. But if they do not look at any of these 4, then it's incorrect, right? So then basically they are looking at blank space. You don't want that to happen. So what you can do is, because it's a saccade, you see there are a range of triggers. First one is a fixation trigger, which means it will detect if the eye is fixating at a certain point. That's not what you want. You want the eye to move and land at a certain point, which is a saccade trigger. So you will choose this one for this particular design, which is a saccade trigger. Yeah. So did you see what just happened?
Cannot be added.
Yeah. So when you click directly, it says it cannot be added. Why? Because Experiment Builder has this function that if only you're recording eye movements, you can use eye movement triggers. Okay. How does it know when you're recording eye movements or not? There is a, where is when you click on the sequence, the head of this file, you see a record option here. You have to tick it. You have to only tick it when you are recording eye movement. You're not doing that during calibration. The PC does it on its own. You're not doing it during instruction. You're not doing it during breaks. You are doing it when the task is running. So in the innermost layer where you are making the task is when you tick record. Okay? You don't tick record at any other layers. When it gives you an error, it doesn't run correctly. So when you tick records, now you can use your eye movement. Now it will not give you that error. Okay? So now I've used the Saccard trigger. It has to connect with the screen because the Saccard is happening on that screen. So the screen will come first. Now I've connected the trigger. Okay? Now let's look at the properties of these triggers. Okay. Alright. So quickly pay attention here because we will have to rush through. What you can try on your own. We will give this particular task as your assignment for the day. So you will all get the paper. It has all the methods in it. Just try to make it as much as you can in your experiment builder files. So we will look at it again tomorrow before the end. Okay. So in the Saccard trigger, apart from the basic features, you have the region from which the Saccard has to get triggered. Like I said, it's a big circular interest area. Eye movement can come from the bottom right or the top left or the top right or anywhere. If you want to specify where the eye movement is coming, only then the trigger will get activated. Only then it will detect it. Then you specify the angle. So there is 0, 10, 20, 30, 45, 90 and then so on. Okay. So this is 0 and then this is 90 and then this is 180 and then 270 and 360. So you specify which region you want. If you want a specific region, if you do not want a specific region, let it select all of it. So the eye movement can come from anywhere. As long as it lands in that area, it will get detected. So once that is done, the type of your interest area, right now it says rectangular, but when you click on it, it gives you a list. So it can be a rectangular shape, it can be an ellipse, or it can be an interest area. So I am selecting interest area because we have made interest areas on that screen. Okay. So as soon as you do that, now it shows you display screen. It will show you whatever screen has interest areas in your program. Okay. Before it was not showing. Now it says display screen because the screen before that, the display screen does have interest area. Just to make it a little clearer, I will rename it to Saccard screen. Okay. So now, do you see here? It says Saccard screen. Okay. So on the Saccard screen, there are some interest areas. Alright, but there are multiple interest areas. You want to be specific which interest area is it detecting. You don't want it to detect everything. So as you click on that bottom column says interest area region, then it shows you a list. It shows you this list of all possible interest areas. It says 1, 2, 3, 4. You can name it whatever, top right, top, bottom, left, whatever you want to call it. And you can select. So let's say the first one I'm guessing is the fixation because I kept it in the center. So let's say I'm selecting the first one here and I'm closing this. Okay. So now the first one shows up here. So any eye movement in that interest area will be detected by this trigger, this particular trigger. So the name of this trigger can be specific to that interest area for you to remember. This trigger only detects eye movement in interest area number 1. Okay. So I'm writing Saccard 1 just for the sake of explanation. Now you know there are 4 eye movements, not 1. 4 potential eye movements. They might make any one of these, but the possibility of 4 exists. So you will add 4 simultaneous triggers for one interest area each. Okay. So now just link the screen to all 4 of them.
We name it accordingly like Saccard 1.
Yes. Yes. So you do the same thing. You click on this. Now instead of rectangle, you select interest area. Then the screen pops out. it says Saccard screen. Now you select number 2 here. You close it. So now it takes eye movements for number 2. You do the same thing for the next one. Interest area. You select number 3. Close it. Number 4. So now we have 4 triggers for 4 potential eye movements. Okay. Now we want the eye tracker. Now it is detecting all 4 eye movements. Wherever it lands, it will trigger the next action. But we want to, while it does that, we want to know where the eye also landed. So do that. For it to do that, we have to use an action trigger, which is like this, x comma y. So this is a very important action trigger. This particular trigger updates values of things during the experiment. What do you need to update during the experiment? Where did the participant look? Was it a specific, the name of that location, the name of the interest area, have they pressed a button or not? Things like that. Things related to the response you need to update during the experiment. If it's a trial, you need to update the trial number during the experiment. So all those things. So what you need to know that these 4 are giving you different values because they are the same trigger. They are getting triggered by all 4 regions. But how to know that? So what you do is you create this x comma y. I'll tell you what to do with it. And you also add 4 of these and connect them to these independent triggers. Okay. So okay, we will just wrap with this one detail because most of it is almost done. Okay, so you just pay attention for this bit and then we can go back to trying the experiment. So when you double click on x comma y, this window pops up. Like I said, this is to update value during your task. Okay, so what do you need to update in this particular situation? Where did the eye movement land? Okay, so there are attributes and values. Attribute will be the name of your variable. Okay, right now you have no variable because you did not create any. But when you create, it will show up here. We will do that right now. And what will be the value that you have to link here? Okay, so I'm closing that. And what you need to do is go to others. And you see this eye icon. Do you see the eye icon?
Yeah.
That's the icon for variables. Just drag one eye and bring it here. Okay. So this is a variable. Call it Saccard location. Okay. This variable will measure where your Saccard has landed. I'm calling it Saccard landed. Now I can call it, let's say I want to name those 4 locations 1, 2, 3, 4. Okay, so I can make this variable a number type. Okay, if I want to call it alpha, beta, gamma, delta, whatever, then I make it a string type. However you want to call it, for you it has to make some sense. Then you make it like that. So right now it's a string variable, because it will give it some value, but I am making it 1, 2, 3, 4. So I will give it a baseline value 0 because 0 means nothing. So it starts with 0 in the experiment, but as it moves forward, it will take the values of 1, 2, 3, 4. Okay, so now it's a number type. It's an integer. So now you see the type has changed here. It doesn't say string anymore. It says integer. Alright. Okay. So you have created a variable. Now this variable has to measure the eye movements landing in a certain region. Okay. You don't have to have 4 variables, mind you. You have to have only one variable. Yeah, only one variable which will have values based on which trigger gets fired. At one point, they will only make eye movement to one location. So at a point, only one of these 3 to 4 triggers will be active and the other will not. So whichever gets active will give value to that variable. Is this clear? Okay. So now you click on x comma y. In attributes on the right hand side, double click. You see a bigger pop-up window. Okay. when you see this pop-up window, you have to go to your, in this field, node selection, go to your variable. This one says Saccard location. That's the variable that I created. Double click on it. Click on value. Click OK. Okay, okay, so I click OK. So now it shows up here. You see, it says Saccard landed value. Okay, so the value of this particular thing will come there now. Whatever values it gets, it has to be associated first. So now you have loaded your variable. You have to load what value it gets. You're not going to specify the exact numbers. You're going to specify where it gets value from. Right? So now when you click on this side, another dialog box opens. You click first trigger. You click the first trigger. This is Saccard 1, Saccard 2, Saccard 3, Saccard 4. You have 4 of them, right? So you click the first one and you click triggered region type or region direction or triggered data. Sorry. You click triggered data and there has to be an ID. Yeah. You see IA ID. You double click on that. Okay. So now that particular variable will be triggered when in this action node, in the Saccard 1 action node, it is triggered and it will report the ID of that variable. You just click OK. Okay. You click OK, they are linked. So I should have technically called it. I just realized we need 4 variables. So we actually need 4 variables. It will.
For every one of those Saccards, you need a new variable.
Yeah. So you can make it, call it as Saccard landed 1, 2, 3 and 4. You just do the same process. Load the variable here, link it to the ID of that particular trigger. So once you have done that, as all of them get loaded, it will trigger a region. That's it. Yeah, so that is about it. As soon as you, yeah. So your experiment is done. As soon as they have made the eye movement, you have recorded where it's gone, your objective is met. Now you want to finally add a result file.
The Saccard landing variable will be just kept like that, like isolated
Yeah.
like that.
Yes, it's standalone. It doesn't have to be connected to anything. So you just click all the remaining empty nodes to a result file from the action box. Okay, it is critical. So there are 2 icons for result. One is in the action box that you use here in the design, but in the beginning of your experiment, in the very first layer, you go to others and there is another result file which looks very similar, but it's missing the plus on the top. So you just drag one and keep it here. That's it. This is just it references the result basically. It references back to the result. That's it. It doesn't have to be connected to anything. So I will just go through what we built so far. First layer has a start, a big whatever window and a result file. This is from the others, mind you, not the action, others. You see what I'm talking about? The categories here on the set. Okay, you double click this. Now we have our calibration. Now we get inside. Then we have instruction. We go further in and yeah, and now before the instruction there was a break and everything in the data source, and now we have the main design. So this is it. This is how your experiment is designed. Once you've made it, you don't have to do much. No piece of code has to be written. You just have to drag it down, link everything up, and it runs on its own. There are just a bunch of good practices to not let the backdrop of this code get corrupt. Once you're done with everything, you save it, of course. You save the file with whatever name that you want to save, if you want to change it or whatever. There is a test run option here. Like I said, if the resolution is same that you want to use and that you have coded in, then you can test run immediately. It will run the experiment. It will give you, right now it will give us a lot of warnings and errors because many things are missing. So the thing about the test running here is we will get to this little bit in the demo when we are running the experiments, but I will just go through quickly. When you do the test run, it shows you some in orange and some in red. So the ones in orange are just warnings. It doesn't mean that your experiment will not run without them. It just means that it is missing some little details that could just make it more clearer and finer. But the red ones are errors that need to be fixed. If you don't fix those errors, the experiment will not run. The good part about this design software is that if you double click on it, it goes back to where the error is. You don't have to find it, which is why I like it so much. So you click on it, it goes back to where the problem is. So somewhere something is missing here. Then you can just reference to what the error says. And
clear input cues को डालना है.
क्या डालना है?
Clear input cues. नहीं तो result file में क्या जाएगा?
कहाँ लिखा है मैंने?
वहाँ पे नहीं. Yeah, it's.
Oh yeah, okay. Yeah, result file has to have something, so you can't clear things. So just untick it. Okay. So that's it. That's all you do. As you get the warnings, you double click and debug it. The SR Research Forum has a lot of support and they are very quick. So if you can't automatically read and understand what is wrong with your design, copy paste the error on their forum, ask them a question, they respond to you immediately. They are very quick with it. If you don't want to do even that, you can just Google it because there are so many people using SR all the time across the world. You will find some answer from it. So debugging is pretty easy and faster. Sorry? No, so when you click on this file, the clear input cues was ticked. The data would not come to the file. You have to untick it. So there are multiple other features that we did not speak about, but they are mostly not needed for a very simple task. The things that you would need are already discussed here. There are some few particular nodes that are specific for certain tasks. When we discuss in our designs, when we are running experiments and we are showing you particular tasks, then we will discuss about those nodes more in detail. Okay. So today what you have to do is this paper will be shared with all of you. Just make the design and come back to us tomorrow in the demo. We will, if you have any queries in making this design and if it doesn't run, if it's not making sense, something is confusing, please ask us. Otherwise, you can just run for yourself and try things out on your own. That's it. Then we will do the.
Do we have a coded view of this whole experimentation? We have the code.
No.
We don't.
Although there is a way to edit, I will just check and tell you. I'm not exactly sure, but right now in this interface, I'm not sure if the demo version allows it, but there is a way to override the code, yes. So that's it. So when you're done, the final step after you're finished with everything is deployment. So do you see this button here with an arrow outside? So that's the deployment. The moment you click it, it packages it into an application file. You specify a location, give it a name, and it will package it. It will only do that if you don't have any errors. There is one here. We will fix it. But there is an error here. No, I